Hello, and welcome back to evolution. Today we want to actually define evolution. What does it mean to say that a population is evolving? So, let's review a little bit. Which factor is the most important in producing the variability that occurs in each generation of humans or any other sexually reproducing organism? Is it natural selection, non-random mating, diploidy, genetic drift, or genetic recombination? Well, it's genetic recombination, as it is in all sexually reproducing organisms. So here we're looking at 3 generations of rabbits, mammals, sexually reproducing. And so in this first generation, you can see that you have 10 individuals. Each of these individuals has 2 alleles, as is the norm. And in this case, this gene has 2 alleles. One is dominant and one is recessive. So this rabbit right here is homozygous dominant. It has 2 dominant alleles. This one is heterozygous. It has a dominant and recessive. And this one is homozygous recessive. It has 2 recessive alleles. And this, of course, is a review. We've talked about this before. Now, when we look at this, we can see that we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 dominant alleles. And we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 recessive alleles. Now, we also happen to have 8 brown bunnies and 2 white bunnies. Well, in the next population, as these guys breed together, we're going to jumble up those alleles, and so there's a redistribution of the alleles. In this population, you can see that we have 2 homozygous dominant, we have 1, 2, 3, 4, 5 homozygous dominant, 1, 2, 3, 4 heterozygous, and then we have 1 little homozygous recessive. And so we've had a shift in the allele frequencies. Now it's 70% of the alleles are dominant and 30% or 0.3 of the alleles are recessive. And so we've had a shift and we're pushing away from this recessive allele. And there's probably a reason for that. If you think about natural selection, what's going to happen to these little white bunnies? If you said they're going to get eaten, you would be absolutely correct. And it turns out that as things go, right, let's say 2 of these rabbits reproduce, there's a higher likelihood now that homozygous dominant rabbits are going to breed with each other than there was before, and so now we're more likely to have the the dominant allele in the population. Now, in reality, probably all these bunnies would breed. In this case, we had 5 of them, whatever. But in this example, we just have these 2. Now, if you throw in this heterozygote here, then you can have an allele, the recessive allele, continue. But that's not what's happening in our population. By the way, this is drift towards one or the other allele. And interestingly enough, over time, many alleles actually do go to one or the other, right? And so the frequencies become 1 and 0. That's not always true, but if there's no actual pressure, sometimes they do, or if there's an intense pressure, sometimes they will as well. Okay, this is an example right here of evolution. This is the very definition of evolution. You have this allele frequency here that's 0.5 and 0.5, but over time you see that it changes. If the allele frequency is changing, then you have a population that is evolving, right? Now, on the other side of this, though, you could have had, let's say all of these rabbits are breeding together. You could have had another generation, the second generation, that was all heterozygous, right? But if you think about having 10 heterozygous rabbits, there are still 10 dominant alleles and 10 recessive alleles. So even though you would have no white bunnies, you would still have the same allele frequency. Your phenotypes change and your genotypes change. But the distribution of alleles, or not the distribution rather, but the number of alleles stays the same. That is not evolution, okay? And we will talk about that more in just a minute when we talk about Hardy-Weinberg. Okay, and so here's just another example of that. So here we have our initial frequency distribution, and we start out, everything is at an even percentage, and then we start to see changes. There's a push away from the yellow that eventually drops. It turns out there's a push away from the blue, right? There's a push away from the pink, and it seems to be a push towards the red. Now here it was actually towards blue, but here we have towards red. So maybe something in the environment changed, maybe it's luck of the draw, who knows, right? But this is evolution. So in a large sexually reproducing population, the frequency of an allele changes from 60% to 20%. This is a hypothetical scenario. From this change, one can most logically assume that the allele reduces fitness, that the allele is linked to a detrimental allele, that there's no sexual selection, that the allele mutates readily, or that random processes have changed allelic frequencies. Well, it seems likely that if you have an allele that was at 60%, the most likely answer, and it jumps to 20, is that the allele reduces fitness. And this is going to be the answer to the mini quiz. Now, having said that, it is also possible that the allele is linked to a detrimental allele, right? Because remember, we have things that are polygenic. And so maybe you have one thing over here that has a huge selective pressure, and it happens to be linked to the allele that you're talking about. In that case, it would also go down, right? And it's possible that random processes have changed the allelic frequencies. That's definitely true. But A and B are the most likely answers, and A is probably, if you're having this drastic a change over a short period of time, then it's probably a direct reduction in fitness. But A and B would be viable, but for the mini quiz, your answer is A. Okay, so do we have natural selection in humans? We've talked about this a little bit before, and I just want to remind you of this example. Skin color was our example. So we know that skin color is actually dictated by multiple genes, and you can see that in having this beautiful spectrum of colors ranging from very light to very dark. And the question becomes, is there a reason for that? And if you look at distributions, you can actually see that when you have more direct sunlight, you typically have more melanin content, and as you have less direct sunlight, you typically have lower melanin content, right? And so there is some benefit if you have direct sunlight to having a higher melanin content, and if you have less intense sun, or less of it in general, having a lower melanin content is more beneficial, right? And so I think that's an interesting thing. So you can see here that you have pushes in certain areas towards melanin content because of the external conditions, in this case being sun angle and sun intensity, right? And the amount of sun per day. Okay, so when you look at a population, we know that the phenotype may change, but the population is not, in fact, evolving. That's just due to natural redistribution of alleles, right? So how do you know that a population is changing? So essentially what we're asking is a population in Hardy-Weinberg equilibrium or not? And I'm going to talk about that, what that means in just a second. But first, it seems that natural selection ought to eliminate variation. I mean, if you think about it, if you have, say, stabilizing selection, and remember from natural selection conversations earlier, stabilizing selection means that there is a push towards the center. If you have that, one would think that you would eventually get to everything being, you know, kind of in the middle. So think about if you had black mice and you had white mice, over time, wouldn't they all be gray mice? Right? And so this is kind of the theory of blending inheritance, and it was a big question, and today we say to ourselves, well, that's silly, because genes are like discrete units. And so you have the genes for color or no color, right? In the case of our black and white, let's say we're talking about black and white mice. You know, and so we think that's silly, but we only think it's silly because we figured it out, because it doesn't actually do this. And two gentlemen, Hardy and Weinberg, independently figured out why you have this genetic variation, okay? So Hardy and Weinberg showed that the frequency of alleles in a population is actually staying the same from one generation to the next, because dominant alleles don't replace recessive ones in the absence of a naturally selective pressure. Even though the genotypes change, the alleles are still discrete units, and that is the Hardy-Weinberg equilibrium. Okay? Now, when we're tracking allele frequencies, a population is said to be in Hardy-Weinberg if the allele frequencies do not change, right? And the assumptions of Hardy-Weinberg, one of the things that helps those frequencies not change, is that you do have a sexually reproducing population. It is a large population because you tend to have rapid changes in allele frequencies in a very small population. Eventually, that was that 10 bunnies. We'd eventually go to one allele or the other just because of random, small numbers, okay? So you need to have a large population. There needs to be random mating, right? Assortative mating, which is the other option. Assortative mating means that the female typically is choosing some trait in the male, right? And so maybe that is large horns, bright colors, big size, right? I mean, think about bison, they're huge. Elephants, they're huge, right? So whatever it happens to be, blue feet, red feet, whatever. So in Hardy-Weinberg, you don't have any pressures at all whatsoever. So random mating, no incoming or outgoing genes, so no new genes, no loss of genes. There's no mutation, that's a no new gene thing. And there's no natural selection at all. So no sexual selection, no natural selection, no inflow or outflow of genes, okay? No changing of genes. So when do we ever have this situation? Like never. So why would these be the assumptions of Hardy-Weinberg? Well, the point is that we're not trying to say, oh, populations are in Hardy-Weinberg. We're trying to say, what does it mean if a population is not changing, okay? And so we use this assumption, the Hardy-Weinberg equilibrium thing, as kind of a base. And if it follows this model, right, then we know that there are no pressures acting on this population. But if the allele frequencies are changing, then there's some pressure that is causing, right, some traits to be excluded or reinforced in a population. Okay, so here's your next question. Which of the following is not a condition for a population to be in Hardy-Weinberg equilibrium? This is just a little review. A small population, random mating, no mutations, no migrations, or no natural selection. Well, if you remember, we need a large population. Small populations are prone to genetic drift. Okay, so all of those other things are true, but we need a large population, not a small population. Okay, so let's talk about the equation for Hardy-Weinberg. First of all, we're going to name the alleles. The dominant allele, which is represented when we're doing genotypes by a capital letter, is going to be P. The recessive allele is going to be Q, right, which is normally the lowercase letter. So if you take all of the alleles in the population, all of P and all of Q, right, the whole population should be P and Q. So those should add up to 1, right? So our first frequency is going to be P plus Q equals 1, right? And so think of it as being all of the dominant alleles plus all of the recessive alleles equal the entire population, okay? Now, we're also going to name the genotypes. If you're homozygous dominant, it means you have 2 dominant alleles. That's going to be PP or P squared. If you are homozygous recessive, you're going to have 2 Qs. That is going to be QQ or Q squared. And if you're heterozygous, that is going to be 1 dominant and 1 recessive. That is PQ. So the equation for genotype frequency, or genotypic frequency, is P squared plus 2PQ equals Q squared. And that equals 1, because it's all the genotypes in the population. And now you're probably saying to yourself, whoa, right? P squared, yeah, I get that. That's this. Q squared, yeah, I get that. That's this. But 2PQ, where does that come from? Because this is only PQ. Well, let's go back to those Punnett squares. Let's say that you can have, if you have this population with 2 alleles, you have P and you have Q. So when you have a distribution of the P's and Q's, you get P squared. You get PQ with this distribution, you get PQ with this distribution, and you get Q squared with just this distribution. P squared plus 2PQ plus Q squared equals 1, the whole population. And that's where that equation comes from. All right, so let's do an example. Let's say that you have a population of 100 jaguars. There are 84 spotted jaguars and 16 black jaguars. Okay, now it's very tempting to say that you have 84 spotted jaguars, and so spots must be dominant, right? So that's something that we would like to look at. But right now what we know is that we have 84 spotted jaguars and 16 black jaguars. So based on the phenotypic frequencies in this case, because color here is completely dominant, we can deduce the frequencies of the genotype. And again, we're trying to figure out, is black recessive? Well, it turns out in this population it actually is. Squirrels, for instance, the black is dominant, right? And so this is one of those cases where the recessive is a smaller number, but let's say that you had a population, you're looking at blood types, the recessive in that case would be a larger number. So for the purposes of our class, we can assume, unless otherwise specified, most of the time the dominant thing is going to be more frequent, but just understand that that's not a guarantee in nature, okay? All right, so let's do this. We have a total population of 100. The spotted population is 84. The black population is 16. If black is recessive, then all of the black jaguars are homozygous recessive. They are QQ, which means that the genotypic frequency for the homozygous recessive, which is Q squared, is 16, the population of black jaguars, over 100, the total population, for a homozygous recessive frequency of 0.16. So Q squared is 0.16, okay? The recessive population over the total population. Now, in order to find Q, because remember, the equation is for genotype frequency is P squared plus 2PQ plus Q squared. Now, for those of you that are excited about doing quadratics and things like that, go for it. But for the rest of you that would like a simpler version, remember the allele frequency is P plus Q. So if you can find out Q, it makes it a whole lot easier to find out the other two genotypes, right? The homozygous dominant and the the heterozygous, yeah? And so we can figure out Q by taking the square root of Q squared. So the square root of 0.16 is 0.4. So we've got Q squared, the homozygous recessive, that's a frequency of the genotype, is 0.16. Okay? And then we also have now 0.4, which is the frequency of the recessive allele. Okay? So now we can take that P plus Q equals 1, switch it around to P equals 1 minus Q, and then we can use that 0.4 that we found out on the last slide to figure out that P is actually equal to 0.6. So now we have Q and we have P. So we have everything we need to know to figure out the homozygous dominant, P squared, which is 0.6 squared, which is 0.36, right? And then we can also figure out the heterozygous, right? Individuals, or the frequency of that, which would be 2PQ, which is 2 times P times Q, which would be 2 times 0.6 times 0.4, which is 0.48, which is our heterozygous, right? So now what we need to make sure is that the genotypes add up to 1. Okay, so we're going to check our work by saying, okay, we know that P squared plus 2PQ plus Q squared equals 1, right? We figured that out. Okay, so now we take that P squared, 0.36 plus 0.48 plus 0.16 equals 1. Yay us, right? Now, we figured out the Hardy-Weinberg equilibrium. So we know that in the next generation, if the allele frequencies are different, right? Because remember, P was 0.6, right? And Q was 0.4 in this generation. If the next generation, no matter what these numbers are, right, as long as the allele frequency is 0.6 for the dominant and 0.4 for the recessive, we are in Hardy-Weinberg equilibrium. If it is not, then we are not. So let's look at another one. Okay, so how about this? Widow's peak, something we've talked about. We know that widow's peak is a dominant allele, right? And then the straight hairline is recessive. Let's say that we have a population of 500 individuals, right? 125 have a straight hairline. We know that straight is recessive, so we can use that number of the homozygous recessives to figure out how many individuals are homozygous dominant and how many are heterozygous. Because remember, this is completely dominant. So while we have 125 that we know are homozygous recessive, all of the rest of these 375 individuals who have a widow's peak, we don't really know how many are homozygous dominant and how many are heterozygous. And this is a way to make a prediction about that. Yeah? And also to determine if we have a population that's evolving, if we have measurements from different generations. So first of all, let's think about what we know. We know that the total is 500. The homozygous recessive is 125. We're going to calculate Q squared first because that's what we know. And Q squared was the homozygous recessive, 125, over the total, which in this case is 500. So our Q squared is 25. So now we can calculate Q, right? And Q is simply the square root of Q squared, which in this case is 0.5. Now we use that to find P. We say P is equal to 1 minus Q. So 1 minus 0.5 equals 0.5. So we have 50% dominant alleles, 50% recessive alleles. So now we can find the other two genotypes by using the p squared equals 2pq plus q squared. So p squared equals 0.5 squared, which is 0.25. And 2pq equals 2 times 0.5 times 0.5, which is 2 times 0.25, which is 0.5. Right? And so we know that homozygous recessive, 25% of the population, homozygous recessive, 25% of the population, heterozygous, 50% of the population. We know that the dominant allele is 50% of all the alleles, the recessive allele is 50% of all the alleles. So now let's use it to determine if this population has some pressure on it. 10 years later, we have a population that's 750, right, because it's gotten bigger. 150 of them have a straight hairline. So the question is this population in Hardy-Weinberg equilibrium, or is there a push towards widow's peak or straight hairlines? So again, let's identify what we know. N is equal to 750. The homozygous recessive is 150, so we're going to calculate Q squared. Q squared is going to be 150 over 750, which is 0.2, right? And then we calculate Q, and we find out that Q, which is the square root of Q squared, is 0.45. Well, this is a big indicator that something is different. So now we're going to find P, and we find P to be 0.55. Well, we know that Q in the last question was 0.5. Well, 0.5 is not 0.45, so is this in Hardy-Weinberg equilibrium? The answer is no. Okay, so Hardy-Weinberg says that allele frequencies will not change, but they do. So which of the following is not true? Allele frequencies often change in natural populations, with some alleles increasing in frequency and others decreasing. The Hardy-Weinberg principle gives us a baseline to measure change. By examining how various factors change the proportions of homozygotes and heterozygotes, we can identify the pressures affecting a given situation. Or all of these are true. All of these are true, okay? All of these are true. Now, let's just review from the other side. We know when Hardy-Weinberg does apply, but let's just go ahead and state when Hardy-Weinberg does not apply. If there's mutation, if you have populations, different populations that have gene flow, if you have very small populations that are subject to genetic drift, if you have non-random mating, sexual selection, assortative mating, inbreeding. Inbreeding is a huge problem for that, and then natural selection. That is when you're going to have changes in allele frequencies. In the absence of those things, you are not going to have changes in allele frequencies. Okay? Now, let's take that one step further, and let's go back to the different types of selection that we had. We had directional selection, which was we're pushing towards one or the other allele. We had stabilizing selection, where we're pushing towards the center, right? So remember, directional is like the cheetahs, right? Faster cheetahs. We're getting faster and faster and faster, get more agile gazelles. So we're getting more agile and more agile. Stabilizing selection, the good example of that was birth weight. Babies that are too big don't come out, and babies that are too small are sick and usually die. Okay, and then we have disruptive or diversifying selection, and that's where we split out the middle, because whatever the heterozygote is has something, that center, right, is no good. So let's say that you have white flowers, right? And you better. Let's say that you have white foxes and you have brown foxes, right? And the white foxes are the homozygous recessive, and the homozygous dominant are brown foxes. But then the heterozygote is gold. Well, maybe that's not camouflaged anywhere, and so you don't have those guys surviving. That's disruptive. Okay, we can translate that to what we're talking about here with the Hardy-Weinberg. In directional selection, one homozygote has the greater fitness. In stabilizing, the heterozygote has the greatest fitness, right? And so a good example of that was the heterozygotes in malaria-prone areas, because the individuals that are heterozygote for normal oxygen transport and sickle cell anemia have greater resistance to malaria. Remember, we talked about this, right? So in that case, the heterozygote has the greatest fitness. That is called overdominance. And if the heterozygote has the lowest fitness, right, they're the ones that are being selected against, this is underdominance. Now, an interesting thing about these three, right, is that the directional selection and the disruptive selection, right, the directional selection removes variation from the population. Okay, so eventually you lose one of those homozygotes, and then you have all of the same allele, right? And that was what we saw in the brown rabbits and the white rabbits in the first slide. Okay, stabilizing selection maintains variation. You'll always have homozygous recessives, you'll always have homozygous dominant, but the heterozygotes will be the ones that are most successful. Okay, so those are the ones that you'll have the most of typically, and so you really have a population that maintains variation, which is how you still have individuals that have sickle cell anemia, which is typically lethal, although there are great treatments for it now. Disruptive and diversifying, it maintains variation, but it's really unstable. These populations will split and become new species, and then you typically have directional selection acting on these split populations. And so you don't have a disruptive selection or a population that has heterozygote underdominance. You don't have that for very long. Typically that will split and speciate pretty quickly. Okay, so that is it for this conversation. If you have any questions, please let me know at the beginning of class, and we will be practicing using Hardy-Weinberg to determine if a population is evolving or not in class. So thanks for listening, and I will see you next time.